Hello everyone, I hope you're doing well. So today's video is going to be about aerobic respiration. So let's start off. There are four steps of aerobic respiration, including glucose, link reaction, Krebs cycle, and electron transport chain. So let's start off with glycolysis. Glycolysis occurs in the cytoplasm of the cell, and there's some steps of glycolysis, so let's undergo that. First of all, in the first step, we've got glucose, which basically uses up one molecule of ATP and undergoes phosphorylation in order to form glucose phosphate. This glucose phosphate undergoes isomerization in order to form fructose phosphate. The fructose phosphate uses another molecule of ATP, undergoes phosphorylation again in order to form fructose 1,6 biphosphate. So this 6C fructose 1,6 biphosphate splits into two 3 carbon triose phosphate. So we got two of these from this one singular 6 carbon fructose 1,6 biphosphate. We got two 3C triose phosphate. The triose phosphate undergoes dehydrogenation, which is basically oxidation, and converts 2 NAD into NADH2, because since there's two 3C triose phosphates, so one NAD for each triose phosphate. And the energy, and this releases energy, this dehydrogenation act, which basically converts ADP to ATP, 2 ATP for each 3 carbon triose phosphate. Moving on, after dehydrogenation, 3 carbon glycerate phosphate are formed, and 2 of these, of course, for each triose phosphate. And these glycerate phosphate undergoes dephosphorylation, basically releasing their phosphates, and again forming 2 molecules of ATP. The ADP plus the phosphates basically forms ATP from the dephosphorylation. So hence, after this entire process, we've got 2 molecules of 3 carbon pyruvate, which brings us to an end to glycolysis. So this was glycolysis. I got a summary table at the end, which will be much more efficient to understanding this process better. So after glycolysis, we've got link reaction, which basically occurs in the matrix of the mitochondria. So inside the mitochondria, link reaction occurs. The pyruvate, which basically enters via active transport into the mitochondria. So only glycolysis was the only reaction which occurs outside of the mitochondria. Henceforth, all reactions will occur inside the mitochondria. In link reaction, you got 2,3C pyruvate, which basically undergoes dehydrogenation and decarboxylation in order to form 2C acetyl, which adds up with coenzyme A in order to form 2C acetyl CoA. And let's just keep this in mind that since there's 2 pyruvate, this reaction will occur twice for each pyruvate molecule. Dehydrogenation forms 1 NADH2 each for each pyruvate and 1 carbon dioxide each for each pyruvate. After link reaction, we've got Krebs cycle, which is also known as citric acid cycle or tricarboxylic acid cycle. This also occurs in the matrix of the mitochondria. So yeah, and this process that I've written here is for the complete oxidation of a glucose molecule, which basically means that 2 acetyl CoA are being used, which is known as complete oxidation, which means 2 turns of the Krebs cycle. If I was only doing for 1 acetyl CoA, that would mean 1 turn of the Krebs cycle. But since it's for complete oxidation, 2 turns will be used for this example. So 2C acetyl CoA is basically formed with oxaloacetate in order to form, which is a 4 carbon molecule in order to form 6 carbon citrate. The acetyl basically merges with the oxaloacetate, and the coenzyme A is reused in the link reaction. The 6C citrate undergoes decarboxylation in order to form a 5C compound and also releases NADH2. The 5C compound undergoes decarboxylation and dehydrogenation in order to form 4C compound, reducing carbon dioxide, NADH2, and also ATP. The 4C compound then undergoes dehydrogenation in order to form another 4C compound, and this time instead of NADH forming FADH2. And the 4C compound undergoes dehydrogenation again in order to form oxaloacetate, once again forming reduced NADH2. So this was Krebs cycle, and since two turns of the Krebs cycle is being followed, all of the products will be multiplied by 2 for each acetyl-CoA molecule. I've also mentioned this here. Electron transport chain are our last step for aerobic respiration. So in electron transport chain occurs inside the cristae of the mitochondria, which is basically the inner membrane of the mitochondria, which is this is a cristae, which is formed in glycolysis and Krebs cycle, move to cristae and release the hydrogen atoms, which basically split up into these electrons and protons. The electrons then move through a series of different electron acceptors or protein carriers on the cristae, moving from high to low energy levels, releasing energy. So the electrons from reduced NAD and reduced FAD are carried by these protein carriers from high energy to low energy, moving from each energy level, and this movement releases energy, which basically allows the H plus ions to be pumped inside the intermembranous space of the mitochondria via active transport. So the energy which is released is used the pumping of these H plus ions inside the intermembranous space of the mitochondria. So because of this high concentration of H plus inside the mitochondria, these H plus ions then move out of the intermembranous space via this stock particle, and in doing so these release energy, electrical potential energy, which is used to convert ADP into ATP, also activating the enzyme ATP synthase, which basically helps in this process. So in order to sum it up quickly, the protons diffuse out using the electrical potential gradient through the protein channels of the stock particles, activating the enzyme ATP synthase, which then finalizes synthesis of ATP using the electrical potential energy of protons. And this process is known as chemiosmosis. The electrons and then protons are then finally accepted by oxygen, releasing water as the byproduct, which is very important because oxygen is a final acceptor of electrons in the entire aerobic respiration process. Also keep this in mind, one reduced NAD releases 2.5 ATP and multiply by 10, which is the total reduced NAD in the entire aerobic respiration, keeping in glycolysis and Krebs cycle, which comes up to 25 ATP. And one reduced FAD produces 1.5 ATP multiplied by 2, because only 2 FAD are reduced FAD are released during Krebs cycle, which forms 3 ATP. Hence the total ATP formed from ETC is known is 28 ATP. The summary, so for I've got a summary table here, which is pretty cool, I guess. So for complete oxidation of glucose, this summary table is formed. Complete oxidation, I just want to make sure this fact, it basically means 2 turns of the Krebs cycle, which means like 2 pyruvates are used and so on, not like 1 pyruvate or 1 turn of the Krebs cycle. So glycolysis, link reaction, and Krebs cycle. The ATP produced from these processes is known as substrate linked phosphorylation, and from ETC it is called oxidative phosphorylation. It is also an important fact. So in glycolysis we can see 0 carbon dioxide are formed, 2 reduced NAD are formed, 0 reduced FAD, net ATP2 and total ATP4. So hence if you may remember, in the glycolysis reaction, 2 ATP were used up for the forming of fructose 1,6 biphosphate. Hence net ATP is 2, but the total ATP formed is 4. In link reaction, 2 molecules of carbon dioxide to reduce NAD, 0 reduce FAD, 0 ATP. And in Krebs cycle we put 4 carbon dioxide, 6 reduced NAD, 2 reduced FAD, 2 net ATP and 2 total ATP. ETC 0 everything else apart from ATP. We got 28 net ATP and 28 total ATP. And if we add them all up, net ATP we can see is 32 and total ATP is 34. So yeah, this was basically aerobic respiration in a nutshell. I will be putting the link for this PDF in the description if anyone needs it. And that is it for today. Hopefully you enjoyed, and if there are any problems, so you can always leave a comment. Good luck and goodbye.